Okay, welcome back. Thanks for being on time. All right, so now we enter the last third of the course. So this part is about, it's going to start with tissue types, and then we're going into organ systems, and that's it. So we're right where we should be in terms of timing, and I'll talk a little bit more about the sort of outline of these last days that represent weeks of lecture. So as I've talked about a lot, you have some important things to bring with you tomorrow at 9:30 or 12:30 for the nutrition lab. They're worth the equivalent of, I don't know, 6 or 7 quizzes, so you don't want to miss out on those things. Does anybody have any questions about the diet diary, the activity log, or the food labels? Yes. That's right. So if you're having like a Subway sandwich, and you could just get the nutrition guide from Subway, or you could look it up on a phone app or something like that. But you wouldn't break it down into there's turkey in here and tomato and stuff. You'd use the whole thing. Anything else? Yeah. Oh, the food. Yeah, you could do the picture if you wanted to. Yeah. Okay. Now that you've just finished your second midterm, and I'm hopeful, but I'm also tired, so I'm not sure if I'll have this done for you tomorrow, but might. So we might get that back in the lab tomorrow, or you might see your grade tomorrow and we could go over it one time in class on Monday. You're reading ahead in chapter 11. And now what I did with the textbook was I tried to, it sort of has big chapters on headings like the digestive system and the respiratory system. And then it has a more compact version, the essentials of those that are all kind of tossed into chapter 11. So you can read both the chapter 11 versions of things and the larger chapters if you just use the downloaded version of things. So there's 2 sets of information that you can look at. I'm asking you to complete text assignment number 10 for Monday. And that's right, we're skipping text assignment number 9. So text assignment 10 is due Monday. Would you make a note of that? That's June the 6th. With the completion of this lecture exam too, you still have 40% of your grade, maybe a little bit more with the lab assignments yet to come, but basically 40% left. You'll have a lab exam in the last week of classes. That'll be on a Tuesday it's scheduled for. So you have this nutrition lab tomorrow. Then next week you do part 1 of lab 10, and then on Thursday you do part 2 of lab 10. Then the following Tuesday is the second lab exam. It'll have the same format as the last one, and it's not a comprehensive exam. Okay. It's time, if you haven't already, to check your final exam schedule on CamLink to find out when and where that occurs. Again, that's completely up to you. And let me just take a moment to outline again what the final exam will feel like. The exam you just took was about 60 points or so, like the first exam. Your final exam will be about 80 points on the last third of the course, the things we're starting on today. And then there'll be 20 points that'll be added approximately from the first third of the course and 20 points from the second third of the course. And I will give you guidance and hints on all aspects of that well before the final exam date. So it is a comprehensive final, but it's also weighted to the last third of the material. Any general or specific questions? Okay. Well, what I hope you'll find in this last third of the course will be the coming together of some ideas that were talked about in the first days of the class. We'll talk about homeostasis, we'll talk about nutrition and food energy, digestion, chemical bonds, all those kinds of things that are now showing up in human or other systems that we're examining. So while the emphasis in this anatomy and physiology part is mainly on humans, we will also compare some respiratory systems and things and digestive systems. So there will be little bits about, fish gills and lungs and cell membranes and things like that as respiratory organs, but it'll be mainly based in human organ systems. Okay. So getting started. Not really dramatically important point, but when we talk about the diversity of life, and this is not a diversity course, the organisms that fall into the category of animals, those are organisms like us that are heterotrophs, that they eat other things, and they tend to have the same kind of tissue types and organ systems to some extent. The simplest kinds of organisms, just sort of for your information, are the asymmetrical ones. Simplest kinds of animals really are the sponges. Moving up from there, you find organisms that have a radial symmetry, a primary radial symmetry. Now your mind is going to sea stars and starfish and things like that. Those are obviously radial symmetry. Turns out, though, it's called secondary radial symmetry because as embryos they have bilateral symmetry. But things like jellyfish, for example, have this kind of radial symmetry. We're really concerned with bilateral organisms, ones that you can kind of split down the middle and have equal sides. Organisms like this have head ends, anterior and posterior. They have digestive systems that have two openings instead of one, like jellyfish do. And we're really concerned with those kinds of organisms most of all. This you can just glance at. When we get into labs next week, we'll talk about these different planes of dissection and different dissection terms. And they will come up more so in the lab than in this portion. Okay. You'll remember at the beginning of this course, or a few weeks in, we started talking about the 4 kinds of macromolecules: carbohydrates and proteins, lipids and nucleic acids. Everything was made in all living systems of those 4 kinds of molecules, and they tended to have similar functions in all organisms. What I want to talk about now is 4 tissue types. So all multicellular organisms are going to have tissues. Now, what's that? That just means certain cell types that are grouped together to do a common function. Maybe a simplest one to think about would be muscle tissue, okay? So your bicep is not an organ, it's a collection of muscle cells all working together, okay? Now, so please write down these 4 tissue types. I'll ask you about these. We're going to go into some detail about each one. And let's start out with the one that will cover the least, and that's going to be nervous tissue. I'm going to spend about 3 minutes on this, in part because you're going to talk about this in your psychology classes and in other classes. So we're going to leave that one and focus on the rest. We'll talk then about epithelial tissue. Epithelial tissue. Nervous epithelial. A third category of connective tissue. Be like ligaments and cartilage, but also includes some surprising things like blood and fat. So we've got nervous, epithelial, connective, and finally muscle tissue. And we're going to pick out one kind of muscle tissue, the kind that you can control, voluntary or skeletal muscle, and I'm going to show you how, in a very crude sense, how that works to contract. But you're going to need to know, and I'll ask you some listing question or listing with examples, these different tissue types and what they do. Okay. One way to describe epithelial tissues is to say that they coat things, like you are covered in skin cells. Skin cells are epithelial tissue, and that they line things so that the tube running through you, that is gut, is lined with epithelial tissue. Your heart is covered with a pericardium, that's epithelial tissue, and the chambers that you'll see in various hearts and heart models in the lab next week are lined with epithelial tissue to make a very smooth, even surface. So write down that epithelial tissue coats things and lines things. Your blood vessels will be wrapped in epithelial tissue, and they'll be lined with epithelial tissue. Now, the different kinds of epithelial that you'd find is going to be named for how it looks. When you scraped out your cheek cells, they were really quite flat, kind of scaly. They're called squamous cells because that means flat. You're going to have ones that are columnar. They look like columns. So the different epithelial cell types are going to be named for what they look like. Some of those here. So there's like your cheek cells. Here's some in the kidney that are called cuboidal. So, examples of epithelial cells named for what they look like. Also named, or they are also functioning to make the glands of the body. We'll talk a little bit about those now. Epithelial cells make up glandular tissue. One kind of gland that you have, or one category, is called exocrine. These are the glands that have ducts. So if you think of sweat glands, they have an opening. If you think of tear glands, they have an opening, the tear duct. You think of salivary glands. I could put my tongue in my cheek here and I can feel a little opening in the side that I would pay attention to when I'm really hungry. I can feel the saliva coming out of that duct. Also, a part of the pancreas has a duct through which a whole variety of digestive enzymes are going to flow into the first part of the small intestine. So exocrine glands, tear, pancreas, sweat glands, have openings. And all those openings are to the outside of the body. And it brings up a point that we'll talk about later, especially in digestion. You think of digestion happening inside your body, and that's true in one sense. But this tube running through you is open, both ends. Okay? And so, although it's in the midst of you, when those glands pour material like salivary juices into your mouth, that's considered to be going outside of you. Okay? Digestion is going to not happen intracellularly, but extracellularly. So exocrine glands have ducts, pancreas, etc., and they are going to exude a product outside the body. You have a whole bunch, a variety of tissues that are endocrine glands. And these are the ductless glands. So they don't have any tube through which the material moves. And you already know how the products that the endocrine glands make are going to leave the cells that make them. And to remind you of that, we talked about this, where the message leaves the nucleus to the rough endoplasmic reticulum, makes a protein that goes to the Golgi apparatus, that's put in vesicles, that fuses with the cell membrane and pops out. That endocytosis, okay, using the endomembrane system, is a means by which materials can be made inside a cell and then exported into what we'll call the extracellular fluid or the interstitial fluid. And once in there, it has the capability of reaching all parts of the body. So you know that what endocrine glands make are hormones. What's the name of the master gland, as they call it, that's in your head? Pituitary gland. The pancreas is also an endocrine gland, so it's both exocrine and endocrine. In the endocrine part, it makes two hormones that have a homeostatic regulatory influence on blood glucose. It makes insulin that's released through the process in the endomembrane system. And once that's outside the body, it gets into the extracellular fluid, it gets into the bloodstream, it goes everywhere where it interacts with cells and allows them to take up blood glucose. So insulin release brings down blood glucose. If your blood glucose goes too low, then your pancreas releases from slightly different cells glucagon. And it leaves the cell in just the same way, gets into the bloodstream, goes to the liver where it triggers the release of stored carbohydrates, breaking down glycogen into the sugar components, into glucose that it releases into the bloodstream, so it raises blood glucose. Okay? So let's talk a little bit about hormones. We're on the topic. There's a variety of kinds. Two that I want you to know of are protein hormones, like insulin, like human growth hormone. And we also talked about another class of hormones that are the steroid hormones, okay? Like estrogen, for example, testosterone. So those are lipid-based hormones. But what does a hormone do and how does it act? So a hormone is released by endocrine tissue through those ductless glands, gets out into the body. How does it influence cells? Well, it only influences cells that have receptors for it. Have receptors, then the binding of the hormone to the receptor is either going to allow that hormone to get inside or send a signal into the cell that's going to change the metabolism of that cell. Maybe it's easier to think about with something like human growth hormone. This used to be one of the rarest molecules in the world. Before molecular biology, the only place you could get human growth hormone was when people donated their brains to science, and they'd find the pituitary gland in these cadaver brains and grind it up and extract the tiniest bit of human growth hormone. And usually that was rationed out to people who had genetic disorders that caused them not to have enough growth hormone, so that without it they would be very, very short. Okay? So this was how you got that stuff. Now, with the advent of cloning and molecular biology, people found rather early on the gene in humans responsible for making this protein. Since the genetic code is the same in all organisms, they could clone that human growth hormone gene into something like yeast and turn that strain of yeast into a little factory to make this, so that it no longer was this rare and expensive protein, and hence people who needed this could solve this. My friend had a pituitary tumor. They went in, cut out part of the pituitary, but it means that she didn't have the means to make human growth hormone, so every morning she gives herself a little stab of human growth hormone, because everybody needs that. Now, you had a lot of it when you were going through puberty and growing dramatically, and that was because it could influence the growing end of your long bones. So before your bones closed off, human growth hormone that you were producing in large amounts would mean that you could lengthen those bones. Now, at a point, you stop growing. And if you are still exposed to large amounts of human growth hormone, either because you're taking it recreationally or you have a condition like giantism. Do you remember Andre the Giant? Okay. If your bones are closed, then the addition of large amounts of growth hormone are going to make, especially your jaw, expansively large. Because your bones can't lengthen anymore, but they can thicken. So anyway, cells must have the receptors, and with that, the presence of those hormones can change the metabolic processes of the cell and influence them, like for bones to grow longer. But not every cell would have growth hormone receptors. Like, it wouldn't make your pancreas cells grow 3 times as big. You wouldn't have growth hormone receptors there. Okay? So there's a little bit of background about epithelial cells in general, what kind of shapes they have, what kind of functions they have, and then about the hormones that might be made in some epithelial tissues. Do you want to ask anything about this? Yeah, I mean, when developed, so he had this throughout his life, right? So that he grew extremely tall, okay, and everything's proportional to that, and then the continuous production of this from an overactive pituitary meant that he also had those signs of giantism, so that, especially that great acromegaly or something is the name for that giant jaw. Here's some epithelial cells that we'll talk about later, nothing to write down here. These are the kinds that will line your gut. Yeah. And so we'll talk about these and how they work and how they increase the surface area so that you can absorb things, especially in the first part of your small intestine. We just have some different pictures, different kinds of epithelial cells that line. Okay. Now the next type of tissue I want to talk about is connective tissue. Different about connective tissue, a couple of important things. It's going to be the presence of what's called a matrix. The cells of the connective tissue are going to make the matrix material. This is easiest to understand in things like cartilage, ligaments, bone. Just remembering, there was some moment in my life, and I can't remember when it exactly was, when I realized that bone was alive. Because when I was a little kid, I thought, well, it's just this dead stuff you're made of. Well, there's not lots. There's cells, they're called osteocytes in bone, and what those osteocytes make is this calcium carbonate matrix, calcium phosphate too, that the bones are made out of. And in things like cartilage, which you may know of if you've ever had tears or rips in it, or if you eat chicken bones, the stuff that's the cushioning material at the end of the chicken leg, that padding stuff is cartilage. And there's cells in there. The cells aren't that common. They're kind of rare. And so you have these few cells, and they make this matrix material. And in that case, it's kind of spongy and cushioning. And in things like ligaments, there are cells in there too, but they tend to be long, stringy, very high tensile strength structures that are going to hold bone to bone, and then finally tendons, which connect muscle to bone. So what these have in common, is that they make a matrix that they tend to have relatively few cells, and that they have rather little blood supply. So when these are damaged in people, these take a long time to heal. Yeah? What have you got? Was it your wrist? Did you lose some? Okay. Was there some? Oh, and so how long did that take to fix? Yeah. So, and it just gets worse as you get older. Okay? So, if you've got anybody blown an ACL, cartilage problems, seriously? Cartilage problems? Yeah, what'd you lose? Okay, how long did that take to fix? Oh, okay. Did they surgically do it, or did they just let it heal itself? So it's still hanging on a little bit. Yeah. That's what, I've got that too. I went for an MRI thinking I just had some torn cartilage and came back. You have an ACL that's hanging by a few threads. Not good news. Huh? Anyway, you have relatively few cells, little bone supply, little blood supply. Okay? So it takes a long time to heal. Now, the material in the matrix of those kind of things like tendons and ligaments and cartilage is a protein called collagen. I want to talk about that. This is really, really a long protein. And the same protein differently arranged in these different substances. So when you think of something like a loose connective tissue, what would that be? Well, if you just like pinch your arm, pinch some relatively loose skin, why that springs back into place is that there's loose connective tissue under the skin. And if you've ever skinned something, okay, it's not that hard. You take a little probe and you can pull the skin back and just kind of wave this stuff through it, and the loose connective tissue kind of separates easily. What you can see in this image is that the collagen fibers are sort of just all over the place. They're just running every which way. And then there's material around them that gives it that sponginess and stuff like that. But in something like a ligament or a tendon, what you do is you line up all those collagen molecules in a row so that they're parallel to each other, and therefore all of them are giving strength in one direction. The cells you see there, the fibroblasts, are the ones that would make those collagen molecules. The cells of the connective tissue make the matrix. But they kind of give up after a while. So we talked earlier about why one gets old and wrinkly, and that was because you're using oxygen, the electron acceptor, and using oxygen means that you're going to make all sorts of free radicals that are going to run around and disrupt molecules and change their shape by stealing electrons. Okay? And where that shows up most clearly is, or in one place, is with the connective tissues of your body. Okay? Oh, I remember, it was a long time ago now, I went in to get my knee checked for something, and the physician checking me out sort of measured my height, said, "Oh, you're 6 foot and 3/4 inches tall." And I said, "No, you're wrong. Which was kind of stupid because he was measuring me. And I said, "No, I'm 6'1 3/4. No, he said, "You used to be 6'1 3/4, and now you've shrunk because the connective material in your spine is getting compressed with time, and so you're getting just this little bit shorter and shorter." And you've seen this in your aged relatives as well. So connective tissue doesn't remake this stuff. And people spend zillions of dollars trying to make connective tissue that resembles what it was like in their youth. And so people will get facial peels and get their face microwaved and do all sorts of crazy stuff. The point of which is either to make your face swell a little bit so that it doesn't look as wrinkly, or in some cases trying to smack those fibroblasts around so they think that it's time to make new collagen. But they generally don't, so those collagen fibers get old, they get broken down, and hence the quality of the skin changes. So here's the picture. There's those fibroblasts. Not many of them, making, in this case, the parallel lines of collagen that would give a particular kind of strength to a functional tissue, in this case, like a tendon. So there's piece like if you were chewing at the end of a chicken bone, that'd be the cartilage part. That material is susceptible to tearing and wearing as well. So here's the picture representing another kind of connective tissue, the bone. In this case, the osteocytes are the ones that will make the matrix. In this case, it's out of calcium phosphate to give bone and to repair bone as needed. Now, here's a surprising one that you wouldn't think of. But fat falls into this category as well. So the fancy name for fat that you store is adipose tissue. Humans spend almost as much money trying to get rid of fat as they do trying to get rid of wrinkles. And I find this really fascinating. So, one of the new methods of fat treatment is called, what's it called? It's not flash freezing, but it's like you go to the human, what's it called? Cool sculpture, cool sculpting, okay. So this is really great. And so here's the idea. If you look this up, it's really fun. The woman, there's some Harvard physician, it really helps. If you got a good university name, you can sell anything. So this woman went to Harvard and became a physician. And what she claimed was that she gave herself a dimple because she ate so many popsicles that she actually froze the fat cells in her cheek. And this gave her the insight that you could get rid of fat by freezing it. So the idea is that you go in and you strap on these kinds of clampy things, and they kind of clamp your fat. And then you just like read a magazine as they freeze these bits. And the idea is that they freeze them just enough so that they damage them. Because what happens to water when it freezes? It expands, right? This is how come it's so stupid, those people who think they're going to live forever by getting their heads or bodies put in cryopreservation. Because if they ever come out, they're just going to be a puddle, because when cells are mostly water, and when the water freezes, it expands and ruins everything. So with the fat ones, they're saying we can freeze these down, they'll be damaged, and then they'll go in and they'll kind of slowly disintegrate, and I don't know what's supposed to happen with all the fat that gets released, because it's got to go somewhere. But anyway, it's nice and expensive so that people get really committed to the process. And then, okay. And so when I'm sitting around, I like try and think of ways that I can get rich. And so 2 summers ago when I first heard of this, I went to they have little belts that have places you could put heating packs in, right? But I put freezer packs in there. And so I made my homemade freezer pack thing. And it's really uncomfortable. It gets really cold. But then, and of course I was too lazy to actually do something about it. And then like a year later, there was something on Groupon just like it for $100 where you could get little fat freezing things to do your home cool sculpting thing. But obviously, I didn't get rich. Okay. And now, the last surprising type of connective tissue is blood. And in this case, the matrix of blood is the watery plasma. So it's not a protein and it's not bone, it's liquid, and it's mostly water. And we'll talk about the other components of blood when we look at the circulatory system and talk about its function. But maybe most surprising that it is categorized as a connective tissue as well. Okay. And so this brings us to the last category of tissue types, and that is muscle tissues. So the first category is the smooth, and it's also called involuntary. This is the stuff you can't consciously control. You're in an interview, you're on a date, and your stomach is making the most appalling noises, okay? Very cool name for this. For the gut noises that you make, it's called borborygmus, okay? And so it sounds really serious. So if you ever have that, you could just tell I'm just having the little borborygmus. Okay. So your gut is lined with smooth muscle. So it's going to push food along. And this will be an important aspect of understanding the digestive system, is that this is kind of a time-sensitive process. You only have so long to extract the molecules that are absorbable from the time you put them in your mouth. By the time they pass a portion of your small intestine, you can't get any more inside you, and this smooth muscle is going to push that material along. So maybe put there like the gut. Then there's cardiac, right? Which of course is the heart muscle that looks different. And the one we're going to pay a little more attention to, the skeletal voluntary muscle. So this is like your bicep, and your triceps, quadriceps, and your calves. These are the muscles that you have direct conscious control over. Talk about a little bit about how they work. So let's dive in there a bit. These are the three kinds of muscle, and now we'll talk about voluntary or skeletal, also called striated muscle because of the sort of stripes that it has in it. I'll show you more pictures, so let's look at that now. So we're going to look at, and I'm going to have you remember this simple structural element called the sarcomere. It is the unit of function. And that anytime you hear something that begins with sarco, think of muscle. So a muscle tumor would be a sarcoma. And inside the muscle cells, instead of having endoplasmic reticulum, the muscle is going to have sarcoplasmic reticulum. We'll talk about its function in just a minute. What do you suppose that is? Okay, now I mentioned it, now I can't hear anything else. Okay, so let's take a look at this. A sarcomere. And I'll just label some parts of it. So the boundaries of a sarcomere are the Z lines. And riveted to those Z lines are these filaments, a protein called actin, also called the thin filament. And if you could look more closely at it, it would look like a twisted, double-stranded pearl necklace. So if I drew this a little bigger, let's just draw it as a single one. It would be like a chain of pearls. And that's going to be glued to the Z lines. No, I'm sorry, these guys right here. So these are the Z lines. And glued to them, all of these lines are the thin filaments. Okay. And those are actin molecules. And then now picture this, because this is going to be round in shape, okay, like a tube. So I've just cut it away so you could see it in cross section here. So here is a molecule that's got little, almost like fish hook, kind of like hockey stick projections off of it, and this is called myosin, called the thick filament. Okay. So think about your upper arm. If you want to bring like a dumbbell or move your hand in this direction, okay, what you're doing is shortening the bicep. And you can do that, depending on your strength, with a various amounts of weight, okay? So you can shorten this muscle. Now, you can't lengthen this muscle with power. So your muscles must work in opposition. If you want to move in with power in this direction, then you've got to shorten the tricep muscle. So all muscles can do, write this down if you didn't know it, is get shorter with power. They have to be paired in order to move the way that you can move. your lower arm. So picture this. From one end of your bicep to the other, there are these muscle fibers. And inside the muscle fibers, there are groups of myofilaments that are end-to-end sarcomeres. So you have umpteen zillion sarcomeres in the bicep muscle. And depending on the electrical stimulation that you send to that muscle can influence how strongly you contract that. And now I want to talk about how that contraction happens and how it works. But it's not me. Okay. Okay. So what's going to happen here, and just to picture this first of all, is that your muscle is going to be waiting to contract. It has absolutely everything that it needs to contract. Okay? And all that has to do is that there has to be the tiniest structural rearrangement in the sarcomere, and then the contraction will happen automatically. And the substance that's going to make the contraction happen is calcium. And so what happens when your brain sends a signal to your muscle to contract is that it gives it literally a little electrical jolt across the membrane. And that membrane jolt causes calcium to come pouring out of the sarcoplasmic reticulum. In muscles, that's what it's there for. The sarcoplasmic reticulum lets calcium out. And the presence of calcium is going to allow the little heads of the thick filaments interact with the little beads of the thin filaments. And notice how they're orientated. They're going to use ATP, and when the calcium's present, they can interact and pull toward the center, and then spring back and pull toward the center. So those Z lines come ever closer together as they can pull in opposite ways, but moving toward the center, so that the Z lines come closer together. Yeah. So this sarcomere is just this unit of function of which they would stretch end to end to end. Okay? So that's how you're going to shorten that whole muscle, is flood the muscle with calcium, and the calcium allows the interaction, and I think it's better shown in a video that I'll call up in a second or show you next time. Now, stay up late at night and you watch bad TV, then you'll encounter these wonderful half-hour-long commercials for things like Dr. Ho's magic muscle toning belt thing, right? And these are true. I mean, these are things that physiotherapists use as well. And what it is that you sit there and eat chips and put this belt on, and it gives a little jolt of electricity across a muscle group, and it causes it to contract. So in physiology labs, if you go there, you'll put these on your arm, and you'll make your finger twitch, and you can dial up the frequency with which the electrical shock happens, and you can twitch and twitch and twitch. And it's remarkable because each time that electrical stimulation that is mimicking what your brain would send to the muscle is causing the calcium to be released, get sucked back in, released, sucked back in, and you can go and go and go and go. And then at some point, your muscle just goes, screw it, okay? I can't figure out where my calcium should be. I'm stopping doing this, and it kind of locks. And that condition is called tetanus. And what's the other name for tetanus when you get a tetanus shot? What are you trying to prevent? Lockjaw, okay? So tetanus means to sort of freeze because your muscle just can't get the calcium in and out that fast. Now there's another phenomenon that happens that no human, here's like a riddle. I just thought of this, okay? There's something that happens to every human that no human has ever experienced. Great riddle. I just thought of it, and it has to do with calcium in muscle. Damn, that's good. You get a star for that, okay? So that you've all seen this in TV shows, right? Where, you know, they're finding out how long ago the person died, and they're looking for rigor mortis. And now rigor mortis is just this stuff, okay? So that a certain period after you die, after at a particular temperature, the sarcoplasmic reticulum in the muscles starts to break down, and it releases this calcium. And this is, you know, how come sometimes, you know, like corpses in a morgue would like sit up and scare the hell out of people, okay? Because they're getting this release of calcium causing the muscles to contract because there's still ATP around, okay? And you get this kind of freezing, and then eventually rigor wears off, and you get that relaxed state again. So rigor mortis is a function of this calcium release causing the muscles to contract maximally. All right, now I just want to here 11. A little better picture of a muscle fiber, and then inside the muscle fiber are the fibrils, and then you can see the sarcomeres next to each other. And so they're aligned in these tubes. And that here's like, like here's the Z lines of one, and the thin filaments and the thick filaments inside there. I'm going to see if this thing works.
Skeletal muscle, also called striated muscle because of its striped appearance under the microscope, is used to move the skeleton. Skeletal muscles are under the direct control of the nervous system and can produce contractions ranging from quick twitches to powerful sustained tension. Individual muscle cells, called muscle fibers, are among the largest cells in the human body. Ranging from 10 to 100 micrometers in diameter, each muscle fiber runs the entire length of the muscle, which may be as much as 35 centimeters or about 14 inches in a human thigh. Each muscle fiber, in turn, contains many individual contractile subunits, myofibrils, extending from one end of the fiber to the other. Each cylindrical myofibril is surrounded by sarcoplasmic reticulum, a complex of membranes forming a network of interconnected hollow tubes. The sarcoplasmic reticulum contains a fluid rich in calcium ions. Deep indentations of the muscle cell membrane, called transverse or T-tubules, extend down into the muscle fiber, passing very close to portions of the sarcoplasmic reticulum. This arrangement of T-tubules and sarcoplasmic reticulum is crucial, as we will see, to controlling muscle contraction. Myofibrils contain subunits called sarcomeres made up of precise arrangements of actin and myosin filaments. Sarcomeres are attached end-to-end throughout the length of the myofibril, and their junction points are called Z-lines. Attached to the Z-lines are strands of actin and two accessory proteins that form the thin filaments. Suspended between the thin filaments are thick filaments, composed of the protein myosin. The alternating thick and thin filaments give myofibrils their striped appearance. Small arms, called cross bridges, extend from the strands of myosin and contact the thin filaments. The actin protein of the thin filament is formed from a double chain of subunits resembling a twisted double strand of pearls. Each subunit has a binding site for a myosin cross bridge. In a relaxed muscle, however, these sites are covered by two accessory proteins, which prevent the myosin cross bridges from attaching to the thin filaments. But when a muscle contracts, the accessory proteins on the thin filaments are moved aside, so myosin cross bridges can attach to the binding sites on the actin subunits of the thin filaments.
And so it is the moving aside of those accessory proteins that calcium's going to accomplish. So that's what it does. Everything's ready to contract. All you have to do is get those things out of the way, and that's what they'll talk about next. Oh. Or not. Okay. And so when you look up up. Okay, how do I make this go away? Oh, right. There we go. And so there's a million other little videos to look up that will show you the more complete story of when and where the calcium interacts with those things. But the basic story that I want you to know, and I will ask you this on your final exam, is the basic structure of the sarcomere, the names of the parts, and the idea of how this contraction occurs.
The 12 steps to muscle contractions. Step 1, neuron action.
I don't like that one. I like this one.
A muscle's contractile units lie within individual
It's a
muscle
child
cells,
narrating
also called
this.
muscle fibers. Each muscle fiber contains numerous bundles of protein filaments. These bundles, called myofibrils, are organized into repeating contractile units called sarcomeres. A sarcomere contains two types of protein filaments, actin filaments and myosin filaments. Each actin, the neurotransmitter, an organelle that consists of a membranous network of compartments. One protein, troponin, lies at regular intervals
obviously
along
this
the
is a
actin
little more
filaments.
complex than we
Another
need
protein
here.
called tropomyosin runs the length of each actin filament. When the myosin head binds to actin, it forms what is referred to as a cross bridge. After myosin binds to the actin filaments, the molecules.
The 12 steps to muscle contraction. Step 7, calcium binds to troponin. Step 8.
All right.
Calcium binds to troponin and causes tropomyosin to shift and expose myosin binding sites.
Which one?
Step 9.
That one?
Yeah, right there.
You've seen this?
I just watched other videos.
It's final exams week, and that means two things. The library is packed, and I'll be writing papers.
Well, hello there. You caught me while I was working out.
Last time I was lifting weights doing a Crash Course episode. To understand how the sliding filament model works, the first thing to keep in mind is that phosphate and the energy from that broken ATP is already stored in the sodium-potassium pumps, for instance. Those pumps are proteins that can accept sodium ions outside a cell, and then they change shape to release them inside a cell. And also, suddenly at the same time, they become able to accept potassium ions. These shape changers are how cells get a lot of the day-to-day job of living done. In a sarcomere, it's calcium ions that change the shape of some of the proteins so that the myosin can finally have its way and grope the actin strands all around it. Then it'll drag those actin strands toward each other, causing the sarcomere to contract. But when a muscle cell is at rest, there are a couple of things that keep this groping from happening. The first is a set of two proteins wrapped around the actin. They're called tropomyosin and troponin, and together they act as a kind of insulation. Let's just continue our middle school metaphor. They're the chaperones that protect the actin from groping. At this point, each little head on the myosin strand has the wreckage of a spent ATP molecule stuck to it. That's ADP and a phosphate, and the energy from that broken ATP is already stored inside the head. So yeah, the myosin has a lot of pent-up frustration. Now, while the muscle cell is resting, it's preparing a stockpile of calcium ions that it will use as a trigger when it's go time. This is done by a specialized version of the smooth endoplasmic reticulum called the sarcoplasmic reticulum, or SR. It's wrapped around each sarcomere and it's studded with calcium pumps. These pumps are constantly burning up ATP to create a high concentration of calcium inside the SR. And of course, whenever you create a concentration gradient, you know it's going to get used. So now we're ready for a muscle contraction to start, but what starts it? Well, stimulus, of course, from a neuron. Muscles are activated by motor neurons, and each sarcomere has a motor neuron nearby. When a signal travels down the neuron to the neuron synapse with the muscle cell, it triggers the release of neurotransmitters, which in turn set off another action potential inside the muscle cell. That action potential continues along the muscle cell's membrane and then flows inside it along special folds in the membrane called T-tubules. When that signal reaches the SR inside the cell, bingo, the SR's channels open wide and let all those calcium ions diffuse down that concentration gradient. The calcium ions bind with one of the chaperones, the troponin, which causes the troponin to rotate around the actin and drag the tropomyosin out of the way, revealing all of those super hot binding sites on the actin. With our chaperones distracted, the myosin, it totally goes to town. It reaches all of those little tiny heads along its length to bind up with the actin, and the excitement of that long-awaited precious contact finally releases the energy that came from breaking that ATP molecule. This burst of energy causes the heads to suddenly bend toward the center of the sarcomere, pulling the actin strands together and shrinking the sarcomere. In millions of sarcomeres, in hundreds of thousands of muscle cells, this is what allows me to, lift my arms. You wouldn't think it would be so complicated. Now, in order for the contraction to stop, you're going to have to tear those two proteins apart, because each myosin head is really comfortable here, snuggling with its beloved actin. So it'll take another passing ATP molecule to attach to the head, which breaks off one of the phosphates to release its energy as soon as they touch. That energy breaks the myosin's bond with the actin and lowers the head, leaving it alone and frustrated once more. So it's weird that the energy from the ATP is actually used to make the muscle relax. But in fact, that's why we get rigor mortis. When you're dead, there's no more ATP to make the muscle relax, and all the calcium ions diffuse out of the sarcoplasmic reticulum, causing the muscles to enter their resting state, which is contracted. But you're not dead yet, so let's wrap this up. When the myosin and actin are being separated, the sarcoplasmic reticulum is hard at work pumping all of the calcium ions back inside it and storing them up for next time. That lets our chaperones come back, the troponin and the tropomyosin retake their positions around the actin strand, and resets the sarcomere for the next impulse to come along. Chemistry makes it all possible, from blasting your guns to my awesome dance moves. Thank you for watching this episode of Crash Course Biology. If you want to go back and look at some stuff, because it was a confusing episode today, table of contents. And thanks to everyone who helped put this together. This one was a doozy, so thanks to our head writer, Blake DiPestino. And of course, Amber, as always, for doing our amazing graphics. If you have any questions, please leave them down below in the comments or get in touch with us on Facebook or Twitter. We will endeavor to answer you, as will all of those extraordinarily helpful people who are not affiliated with us at all, but are quite smart and helpful. So thank you to them, and we'll see you next time.
That was kind of nice.
Yeah, he has a bunch of videos that line up great with your course.
Oh,
You can
nice.
follow them through.
Wonderful. All right, so we're all done for today. We'll see you tomorrow at 9:30 or 11:30, and perhaps you'll get your exams back then.